This is a diagram of transcription. First, the RNA polymerase binds to the promoter.
So the next step in the transcription process is initiation, in which the RNA polymerase binds to a promoter and the DNA strands unwind. This allows the RNA synthesis to start on the template strand.
So this is the process of initiation. First you'll see we have a TATA box, a nucleotide sequence in which the transcription factors will bind onto. These transcription factors allow for RNA polymerase to bind and form an initiation complex. So we have the RNA polymerase come in here, and then the DNA strands become unwinding, allowing for an RNA transcript to come in.
As polymerase moves down the DNA strand, the RNA ends up elongating. It moves in a 5-to-3 direction. After it's fully elongated, termination happens and the RNA transcript is released from the polymerase. The polymerase then detaches. The 5' and the poly A tail are important because they help facilitate the export of the mature mRNA. They help protect the mRNA from degradation by the hydraulic enzymes. And once the rMNA reaches the cytoplasm, both structures help ribosomes attach to the 5' end of the mRNA.
So this pre-mRNA is going to turn into mRNA, by which introns cut out and exons splice together, like this, creating one large coding segment. So now we have the process of the initiation complex. After leaving the nucleus, mRNA attaches to the ribosome. Here is a ribosome which we made. So you're going to have, a ribosome is made up of two subunits, a large subunit and a small subunit. And then you have binding sites called EP&A binding sites. What binds to these EP&A binding sites is tRNA. tRNA like this with...
Next is amino acyl tRNA synthase. What this does is allows each amino acid to attach to its proper tRNA with help of a specific enzyme and ATP. The amino acid binds to the complex. Then ATP is added. Once this is added, ATP loses two groups. Well, this stays here. tRNA then is attached. This allows tRNA to attach to the tRNA molecule. The two phosphate groups go away, and the tRNA with the amino acid detaches.
Here's the process of translation. Here you have the ribosome again and the tRNA, which are binded to the eRNA sites, and your mRNA. So as the mRNA goes into the ribosome, you're going to have it being coded, each one individually. AUG goes to UAC, AAA goes to UUU. And as it moves along, more tRNA are going to come in, doing this like that. So what this does is allow for the peptide bond formation to happen. So you have peptides like GLY can now go to the top and be a part of this polypeptide chain.
As the tRNA strand moves down the ribosome, eventually a stop codon falls into the A spot. This stop codon is AUU. This allows the rest of the tRNAs to release, allowing a free polypeptide, and this works in release factor and detaches.